Project 3, antigen capture, presentation, and recognition, and adaptive immunity. My name is Alondra Curiel.
I'm Diego Castillo. I'm Roberto Blanco.
What is an antigen? An antigen is something that causes an immunological reaction, more specifically, the formation of antibodies. On the surface of pathogens like bacteria, viruses, or fungi, or even cells from another person, for example, transplants, this can also mean a protein, polysaccharide, or other molecule. Epitopes are a unique portion of an epitopes are a unique portion of an antigen recognized and bound by an antibody or a T cell receptor. They can sometimes be referred to as an antigenic determinant. Multiple epitopes on a single antigen enable different antibodies or immune cells to identify it. Antigen recognition, BCR versus TCR. While B and T lymphocytes are vital for the immune system's ability to identify and react to antigens, there are significant distinctions between how these cells achieve this. The primary cause of distinction is the type of antigen receptors they have, like TCRs and BCRs. BCRs are antigen processing or presentation that isn't necessary for B lymphocytes to identify and stick to intact or natural antigens. The membrane-bound immunoglobulin, known as the BCR, is capable of identifying a wide range of macromolecules, including proteins, lipids, and carbs. TCRs are process antigens presented by major histocompatibility complex, or MHC molecules, on APCs. This includes B cells, dendritic cells, and macrophages. TCRs, as compared to BCRs, mainly identify peptide fragments derived from whole proteins. Unprocessed antigens in their natural state are recognized by BCRs. Proteins, carbohydrates, lipids, and nucleic acids are examples of these antigens. Only processed peptide antigens presented by MHC molecules are recognized by TCRs. They're very specific for fragments produced from proteins. MHC, also known as major histocompatibility complex, are important molecules for the immune system. MHC molecules present antigens to T cells, helping to create an immune response. There are two types, class 1 and class 2. Class 1 is found on all nucleated cells and presents to CD8 T cells, which triggers infected cell destruction. Class 2 is found on APCs and presents extracellular antigens to CD4 T cells, which activates immune responses. Cross-presentation is when APCs take up extracellular antigens and present them on MHC class 1 molecules instead of MHC class 2 molecules. This is because CD8 T cells can recognize and attack infected or cancerous cells even if the APC itself is not infected. Cross-presentation is important for things like vaccine responses and antiviral immunity. This makes sure the extracellular threats can still be targeted by cytotoxic T cell responses.
What is the role of dendritic cells in capturing and presenting antigens to T cells? Dendritic cells are usually exposed to the external environment, so they are usually located on the skin and other mucosal surfaces. When they capture antigens, it is through phagocytosis or endocytosis. Once captured, they begin to process them into small fragments, which are then presented to MHC molecules. After the antigens are processed, then the cells begin to move to the lymph nodes with the use of chemokines. The antigens are then presented on the surface MHC molecules to T cells, allowing for them to be activated. There are two types of T cells that can recognize the antigen: CD4 plus T cells, which recognize antigens presented by MHC class 2 and initiate T helper cells, and there's CD8 plus T cells, which recognize MHC class 1 presented antigens and initiate cytotoxic T cells. So where does the development of lymphocytes begin? So lymphocytes are primarily developed in the bone marrow and thymus. All lymphocytes originate from the hematopoietic stem cells in the bone marrow and differentiate into common lymphoid progenitor cells. B cells will develop in the bone marrow by undergoing a series of maturation steps, including the rearrangement of immunoglobulin genes, allowing for the production of unique antibodies. T cells migrate from the bone marrow to the thymus and undergo development. They have positive and negative selection so that they can recognize antigens presented by MHC and don't react to self-antigens. What type of selections take place during different stages of maturation? B cells undergo positive or negative selection in the bone marrow to ensure they are healthy and self-tolerant. Positive selection ensures that B cells are successfully rearranged in their genes, which produce functional BCRs. If they fail to produce a functional BCR, they will undergo apoptosis. Negative selection eliminates the B cells that bind strongly to the body's own antigens, preventing autoimmunity. It is mainly known as central tolerance. Once both go through positive and negative selection, they have peripheral selection, where they leave the bone marrow, and if they react to self-antigens, they will be rendered as non-responsive or undergo apoptosis. In T cells, they also undergo positive and negative selection. However, there are some differences. Positive selection in T cells takes place in the thymic cortex, and T cells that can bind to self-MHC molecules moderately are chosen to survive, ensuring that T cells can recognize antigens when they are eventually presented by MHC molecules. Negative selection occurs in the thymic medulla, where T cells that are too strong in their reaction/binding to self-antigens presented by MHC molecules are either killed or eliminated, preventing autoimmunity. Peripheral selection is the same, where if they react too strongly, they are either non-responsive or killed.
Okay, what is the role of negative selection during the maturation process? Negative selection during maturation eliminates self-reactive T and B cells that recognize the body's own antigens too strongly. This prevents autoimmune diseases by ensuring only non-self-reactive immune cells survive. Occurring in the thymus, T cells, and bone marrow B cells, it maintains self-tolerance and immune system balance. How and why is the vast diversity of receptor structures in the lymphocyte repertoire generated? Lymphocyte receptor diversity is generated through the BG recombination, where variables B diversity is D and J is J. Gene sequences randomly recombine to form unique receptors. Junctional diversity further enhances variably by adding or deleting nucleotides at segment junctions. Combination diversity arises from different pairings of BCR or TCR. In B cells, additional diversity is introduced through somatic hypermutation after antigen exposure. This vast diversity allows lymphocytes to recognize millions of antigens, ensuring broad immune protection, preventing pathogen escape, and maintaining self-tolerance by eliminating self-reactive lymphocytes during maturation. That's it. Thank you so much.